Hey guys, so this is just going to be a new lecture on membranes, and let's get started. Basically, we already talked about, the introductory parts of membranes, but we still haven't really talked about, transmembrane proteins, lipid rafts. We haven't really gone into great detail about that yet, so we're going to do that next. So let's begin, and the first thing that we're going to talk about is the asymmetry that's involved in phospholipids in membranes. So we already know that phosphatidylserine and phosphatidylthalamine, those are on the those are on the inner leaflet. So the one thing that you have to remember is there's another very important phospholipid. It's called phosphatidylnositol, phosphatidylnositol, and it's very important for signal transduction on the inner side. And you also know that glycolipids are always on the outside, so they're always on the non-cytosidic side of the membrane, because that's how they're made, and you will understand that later. And then we talked about transmembrane proteins. You know that as they're inside the membrane, they're amphipathic, they have a lot of modifications. So integral portion of the protein is, so the integral portion is the portion that's inside the membrane, and then there's peripheral portion where no portion is in the membrane, and all of that. So remember that for future reference. And then we have alpha helix. These are transmembrane segments of the protein, and they're often in there. We also have beta sheets. They can also be integral membranes in the protein. It can be single pass or multi-pass. We also have beta barrels. They form, we already discussed that beta barrels are very important to form pores for pouring and all of that. So beta barrels is always in the middle, and in the middle is this aqueous solution inside the beta barrel, and it forms a pore. So the hydrophobic polar side chains, they always stick out and they interact with other things. So the polar amino side chains face the inside, and the hydrophobic non-polar amino side chains, they face the outside and they interact with other lipid bilayers. So this is something that's very commonly found in bacterial, mitochondrial, and chloroplast outer membranes. So just remember porins for beta barrels. Peripheral, we already talked about, is the ones they don't interact with the lipid part of the membrane. They're not inside. And they're also bound, remember this, they're also bound by non-covalent interactions. And we know these non-covalent interactions as, ionic bonds, hydrophobic force, hydrogen bonding, and van der Waals interactions. So just remember that. So that's very important. And then we're going to now talk about, there's different cell biology methods for predicting protein structure. So there's many different kinds. The four that we're going to talk about is the hydropathy plot, the freeze fracture, liposome generation, and detergent solubilization. So that's going to be in the next video, and we'll get right on to that. Okay, bye.